This lecture is all about taste. We're going to start at the beginning with your primary sensory neurons in the gustatory system and talk about how it detects different tastes, chemical senses, and how that information gets passed on to the rest of your nervous system. So starting at the beginning, let's talk about taste receptors. So they are, as you may expect, located in your tongue. So if you zoom into your tongue and look at it under a microscope, you'll notice that it has all of these little bumpy structures. If you zoom into the bumpy structures, you'll see individual taste buds. And inside each taste bud is a bundle of cells that contains all of your taste receptor cells, TRCs. The middle of your taste bud has a hole through which the taste pore allows chemicals that are dissolved in the chemical environment, like whatever you're eating or drinking at the time, to pass into and have access to the TRCs. And these taste receptor cells will then detect whatever chemicals they will detect and pass that information on through the gustatory nerves that goes through your tongue and projects towards the rest of your nervous system. So let's talk about these taste receptor cells and zoom in for a little bit more. Okay, these taste receptor cells are super cool, and this architecture that you're seeing here should not be a surprise to you because in terms of the organization, these local primary sensory neurons that then project to some kind of ganglion cells that project to the rest of the nervous system is exactly what we saw in the retinal system too. It's a little less complicated because there's fewer layers of cells in between. It basically just goes between the primary sensory neurons and the ganglions. In the case of the retina, we have the photoreceptors, a bunch of different bipolar cells and amacrine cells and horizontal cells, and then they get passed on to the retinal ganglion cells. And so the computation that goes on in the retina is substantially more sophisticated and complicated than what's going on here in the taste system, but it's sort of a similar idea, right? Just a little bit simpler in this way. So we have our primary taste receptor cells, and the taste receptor cells don't actually have any axons themselves. They detect taste, like sugar, for example, or bitterness, or whatever it is. We'll talk about exactly how that works later, the molecular basis of it. But regardless of whatever they detect, they're going to then activate by releasing neurotransmitters at the base to activate the gustatory ganglion neurons. And these ganglion neurons, just like the retinal ganglion neurons, are the only ones that project to the rest of the nervous system. So nothing about the primary taste receptors themselves is ever actually passed on to the nervous system. It's only through activation of the ganglion cells in the gustatory system are they passing information on to the rest of your nervous system. Funny fact about these taste receptor cells, we know ATP, as biologists we know ATP well because it is the primary way in which the cell uses energy and passes that around and uses to power a lot of the cellular machinery in our bodies in various different cell types. In this particular case, taste receptor cells actually use ATP as a neurotransmitter. It's kind of a funky neurotransmitter to be using at this moment, but if you think about how, well, this is a preview, but your umami taste receptors are actually activated by glutamate as a ligand. So when you have glutamate in the environment, it tastes savory to it because that's one of the amino acids. So it may not be that much of a surprise that you can't actually use glutamate as a neurotransmitter when your receptors are trying to detect glutamate in the environment. So here they use ATP. Okay, kind of a cool fact. So I don't know about you, but I remember distinctly when I was in elementary school, I had to do an exercise in science class one day where I had to color something of this kind. There was a map of the tongue, and I had to color in the different parts of it that were responsible for the different tastes. Now, I thought this was true, right? For years I thought this was true. So you can imagine how angry I was the first time I learned that this was in fact not at all true. It doesn't work like this at all in the gustatory system. In fact, each of your taste buds, each of your little taste buds contains receptors for all 5 of the chemical senses. And so the localization of your taste buds does mean that different parts of your tongue are differently sensitive to taste, but there's absolutely no evidence that there's localization of different types of tastes in different parts of your tongue. So I know people who definitely claim that there's a right way of eating certain types of food. You have to put it in different parts of your mouth. Maybe that has to do something with texture or with the flow of air or whatever else it is. I know it is not because there is localization of different types of gustatory receptors on your tongue. It just doesn't work like that. But let's talk about how that does work. So here are your gustatory receptors. I'm only showing you 4 of them. We'll get to the 5th one in a little bit because it's kind of a special case that I'll talk to you about separately. So we have umami, sweet, and bitter. And these 3 senses are mediated by, not surprisingly, GPCRs. Okay? G protein coupled receptors rear their heads once again because they are super duper important. For a view of what they are, you can go back to the lecture that we had earlier on metabotropic postsynaptic receptors. That's where we introduced GPCRs for the first time. And we also saw GPCRs in the vision lectures because rhodopsins, the proteins that are responsible for detecting light and photons in our visual system, are also GPCRs. So it's not really surprising that they're rearing their head here again, because it's something that we see all the time in all different parts of not only our nervous system, but the rest of the cellular and molecular biology of our bodies as well. So umami detects kind of savory tastes and sweet detects sweet tastes. And so both of these are thought of as attractive gustatory receptors, because usually you're seeking food that tastes good. You want something that has high caloric content and isn't going to kill you. Bitter receptors, on the other hand, are GPCRs that usually have the opposite effect on your body. They are aversive for most mammals, and it's because they tend to be things that are toxic and poisonous to your body. So, for example, there are things like quinine, strychnine, and atropine are all perceived as being bitter because we have bitter receptors in our tongues that are binding to them. So, let's first talk a little bit more about the umami and the sweet receptors. They are GPCRs, and there are actually 3 genes that form 2 different types of protein heterodimers that underlie your sense of umami and your sense of sweet. Your umami taste, like I said earlier, is things like that are protein rich, and so glutamate is one of the amino acids, and so you might want to have a taste receptor that is sensitive to glutamate, including monosodium glutamate, MSG, to make foods taste more savory. Now, the umami receptor is a heterodimer of T1R1 as one of the components, and T1R3 as the other component. If you instead take T1R3 and glue it together with T1R2, you have a sweet receptor. So these 2 genes, these 3 genes that form these 2 different receptors, are closely related, and they are able to form different configurations with each other to underlie your sense of umami and your sense of sweet. Now, your sweet receptors are sensitive to things like sucrose, fructose, and a couple of other types of sugars as well, like lactose, for example. There's a couple of different other naturally occurring molecules that bind to sweet receptors, as well as a variety of different artificial sweeteners that are engineered to bind to these sweet receptors as well. Okay, so now that we have talked about these 2 closely related gustatory senses and the genes that underlie them, I want you to do a thought experiment. So here are 2 animals. Here's a tiger and a panda. They're both of the order Carnivora. And so the question is, if you had to guess which taste receptor is missing in which of these animals, given what you know about their food habits. Okay, so you can think about it for a second if you want to. I'm going to keep talking. So tigers are carnivorous animals. They don't eat anything except meat. Okay, so they don't really have a taste. They don't have a taste for sweets. And so, in fact, if you look in their genome, you will find that tigers have lost the sweet receptor gene. They don't have them anymore. Pandas, on the other hand, even though they're carnivorans, have lost the taste for meat. They are vegetarians. They only eat bamboo, and so they, in fact, have lost their taste receptors for umami. They just don't have a taste for umami anymore. They do actually have a ton of extra receptors for bitter, because that's something that's relevant for their dietary habits. So this is a lovely story in convergent evolution as well of red pandas and giant pandas. They are evolutionarily distinct lines here, and you can see that even though they're not related to each other, they have independently evolved a mutation so that they no longer have their umami taste receptor. They have become these pseudogenes that are no longer active in both red pandas and giant pandas. So this is a really lovely paper here, because not only do they have this convergent evolution in their umami taste receptors, there's some anatomical convergent evolution as well, because of their dietary habits, and that's something that ties together a lot about our knowledge of sensory neurobiology, as well as the evolutionary niches and the habits of different animals. So this all kind of makes sense if you take it together. So let's move on then to the bitter receptors. Bitter receptors, as I said, are GPCRs as well, but unlike the umami and the sweet receptors, which is one umami taste receptor and one sweet taste receptor, there's actually dozens of different bitter receptors. And it's because there's just a variety of different compounds in our environment that you may consider not eating. So if you eat something, you chew on it, 2 seconds later, it's like, oh, that tastes kind of bitter. You still have time to spit it out, right? And so there's a variety of different compounds, and so we have actually a variety of different bitter receptors that are sensitive to them. However, unlike sweet and umami, it doesn't actually matter in terms of what I do with that piece of food that tastes bitter to know what type of bitter it is. And so these T2R genes of which humans have 30 of them, have a much lower threshold. So my threshold for tasting bitter is much lower than sweet. I can taste tiny amounts of it, so I can spit the food out really fast if I think it's going to be bad for me. And they're also co-expressed on single taste receptor cells, which means that it doesn't kind of matter which of these receptors is being activated. All I know as the rest of the nervous system in order to guide my behavior is that something tastes bitter. I don't know what type of bitter it is, and it doesn't matter because what I'm going to do about it is I'm going to spit it out, right? So we talked about this in the intro lecture on sensing and action and thinking about the organization of sensory systems, because all that really matters if you think about the goal of the nervous system, why you have a brain and nervous system in the first place, is to guide your behavior and to guide your decision making in the future. So any information that is irrelevant for what you're going to do next as an organism tends to be somewhat ignored, right? So we saw this in the visual system when we were talking about contrast and on and off cells that are in your visual system, like the retinal ganglion cells, as well as the oriented bar cells that are in V1. So they don't actually tell you what's going on. They filtered reality in such a way because all the rest of it doesn't matter. There's contrast enhancing cells because you want to pay attention to what's changing in your environment rather than what is actually there, because that's just too much information. I don't care. This is a similar concept that we're seeing in the bitter receptors. There's basically a convergence where it doesn't actually matter which type of bitter it is. What matters is what I'm going to do about it, spit out the food. And so we have all of these different T2R genes that are expressed in the same taste receptor cells because it doesn't actually matter, okay? So bitter is usually aversive because you have all of these horrible compounds with terrible names that you definitely don't want to eat, like strychnine and quinine and atropine. However, humans, because we're very creative creatures, have actually learned to really enjoy bitter in limited contexts, and this is very much a learned behavior. Usually kids don't like bitter foods, but we can learn to enjoy eating and drinking different types of bitter substances. So the story about sour is also a fascinating one. So we're moving on to what we've talked about: umami, sweet, and bitter. The sour receptor, as far as we know, is not a GPCR. And when I looked up the molecular basis of a sour taste reception in humans and in mammals, I was shocked to realize that it was only in 2019 was the receptor, the molecular receptor for sour taste in mammals, conclusively found. Right? You should just think about it. Like, how long have we known about photoreceptors? How long have we known about touch sensors? All of these things. It's absolutely amazing to me that our knowledge of the gustatory senses is so fundamental to how we interact with our environment every day, and yet there's so much that we still don't know about them. So as far as we can tell, this is the latest that I've been able to find in the literature. The sour taste receptors are a class of molecules that are called otopetrins, and they're cation channels. Maybe not surprising. So this is not GPCR, but actually a cation channel. Now, the otopetrins were previously identified to be involved in gravity sensing in the vestibular system of the inner ear, and that's why they were previously found. And now they've been found in the gustatory system to be expressed in taste buds. And so there's a beautiful set of experiments where it was demonstrated that they were not only necessary but sufficient for evoking sour behaviors in a mammalian system. I think that's really cool. So here is one way you can hack your gustatory system. There is a fruit that is known as the miracle berry. It looks like the following. It's this little red berry, okay? And it contains a molecule inside, which is a glycoprotein by the name of miraculin. Here's its molecular structure. What miraculin does is it has a really funky, it gives you a gustatory illusion, basically. It's a gustatory hallucination. If you chew on the berry for the next half an hour or so, anything that would usually be sour actually ends up tasting really sweet. I'm going to say that one more time. If you basically suck on a lemon after you chew on the miracle berry and then you suck on a lemon is usually quite sour, okay? It tastes like the best lemonade you've ever had. So what happens at the molecular level is that this miraculin, this glycoprotein, binds to the sweet taste receptors in your taste buds. So it binds to those T1R1, T1R3 heterodimers. And then in the presence of acid, it activates the sweet receptors. That's what this glycoprotein does. It's a really funky sensory illusion. I think it is actually possible to buy this in a pill form. So if you don't have access, if you don't happen to have access to a friend who grows a miracle-berry tree, it is possible to acquire this molecule in kind of a pill form, I think. So that's kind of a fun experiment to do if you wanted to do that as a party trick. Okay, so we talked about umami, sweet, bitter, and sour. Now we've come to sodium, which is perhaps actually, despite the fact that it's just salt, and salt is life, actually rather mysterious. It's still unclear to us as neuroscientists what is the conclusive fundamental basis for salt tasting in your gustatory system. We don't actually know what the molecules are involved are. And part of it that makes it kind of complicated is that umami and sweet are appetitive. Bitter is aversive, although outside 10, you can learn to like it. Sour is a little more complicated, and so is sodium. Sodium is complicated because at low concentrations, salt is very much appetitive. If you've ever had food that was undersalted, it doesn't taste very good. If you add just the right amount of salt, it tastes great. But at even higher concentrations, salt is very much aversive. You know, if you've ever gone swimming in the ocean and snorted up a bit of salt water, it's pretty horrible. Like, salt is not unambiguously positive. In higher concentrations. So we do know that there is a set of cation channels that are expressing mice, these epithelial sodium channels, and they are required for mice to taste salt. However, we know this doesn't quite explain everything because for humans, at least, our tongues don't express this protein. So we don't really know what's happening in humans and our ability to taste salt. Furthermore, we don't know what the molecular mechanisms are for salt-aversive properties at higher concentrations. Are they mediated by the same channels? Are there different channels? Are there different systems that are involved? Is this a computation that happens after the tongue? These are all questions that kind of still remain unclear in neuroscience and remains to be figured out. I also will point out here that beyond these 5 different gustatory receptors, people also think about other types of taste and sensory information that you can gather from your mouth and your tongue that are not on this list, like the taste of water, for example, or even the smell of water. There's a couple of papers floating around, and people are increasingly starting to work on these things. And you can imagine it's really difficult to work on something like, what does water taste like? But water, in fact, does taste like something, right? It's not just the stuff that's in, and there's a taste of water. Really hard to study. But I'm telling you here, at least what we do know about the umami and the sweet receptors. The bitter receptors are pretty well studied. The sour receptors, I mean, we're sort of figuring it out. Sodium, we're still working on it. These are very key to our sense of what foods taste like, and at least what it tastes like when we put it on our tongues. Okay, the other thing that we really enjoy in food are things that are actually taste receptors. This is something that is I personally enjoy. I like spicy food, and so spicy food is foods that have this molecule called capsaicin. Capsaicin is this class of molecules that are this particular molecule that's made by different types of chili peppers, for example. Now, chilies produce capsaicin, and capsaicin tastes spicy. We say it tastes spicy. We use that word, but it doesn't actually hit any of the gustatory receptors. It hits something else altogether, which is a TRPV1 channel. And TRPV1 channels are cation channels that are actually found all over your tactile system in your somatosensory system. So we'll get to that a little bit later in the somatosensory lecture. I'm just previewing it here because when you taste spicy food, it's not hitting any of the taste receptors. It's actually hitting this somatosensation receptor. And the TRPV1 channels in particular is one that's usually activated by high temperature. Now, if you know Chinese food, you'll also know that there's actually a separate word for things that are not actually hot spicy, but also numb spicy. This is ma and la. Okay? Numb spicy is mediated by an entirely different, unrelated molecule that doesn't look anything like capsaicin. It's a molecule that's known as alpha hydroxyxanthol. Okay. And this molecule is not made by these capsaicin plants, but is made by this Sichuan peppercorn tree. That one's mine, actually. I used to have a big one in my front yard. I love this thing. And it actually activates, just like the TRPV1 channels activated capsaicin, it activates tactile sensors that are sensitive to vibration. So it's a couple of papers talking about the molecular mechanisms of exactly what is the difference between the sensors that are activated by the numb spicy receptors, the ma receptors, and the la receptors. Okay, so you might be asking yourself, this is kind of an interesting evolutionary question here, because animals like to eat fruit, and fruit make different yummy things for us to eat because they want us to do them the favor of dispersing their seed. All right, so this is why a lot of fruits make fructose, and fructose is sweet, and we really like fructose, so we eat it, and we eat these fruits, and we do them the favor of seed dispersal. So that makes sense for most fruits, but that does not explain why fruits of the capsaicin branch make capsaicin, because it's actually quite aversive in high concentrations. You can, again, as humans, apparently we can learn to like anything. We can learn to like pain, but it is painful. And to most small animals, capsaicin is aversive. So why, as a fleshy fruit in the capsaicin family, why would you bother to make something that make your fruits less appetitive for small animals to eat? The interesting story here has to do with the difference between small mammals and birds and how we eat fruit. So here's an experiment where what they did is this was really a clever experiment. What they did was they compared 3 different types of animals. So it's a cactus mouse, a pack rat, and a thrasher, a type of fruit-eating bird, and they fed them 3 different types of fruits: the hackberry, non-pungent chilies, and finally pungent chilies, chilies that are quite spicy. And what they found is that the small mammals avoided the pungent chilies, but the birds ate all 3 in equal concentrations. So birds will eat a spicy chili just as much as they would eat a blueberry. They really don't care, they like them all. Whereas small mammals, like a cactus mouse or a pack rat, are actively avoiding the really spicy chilies. So capsaicin chilies is now understood to be a direct deterrent. And so the avian TRPV1 channel is insensitive to capsaicin. Birds literally can't taste spicy. They don't have the receptor for it. Their TRPV1 receptor is something else, and it's not sensitive to capsaicin. And the reason the chili pepper did this is the following. It's because small mammals, when they're eating a fruit, will chew it up. We chew it, right? And then for the capsaicin plants, when a small mammal chews up their plant and then chews up the seeds, it is no longer able to germinate when it is dispersed at a far location. Whereas birds will swallow the whole thing whole, and so birds are actually excellent at dispersing capsaicin seeds. And so the chili peppers evolved capsaicin as a direct deterrent. It can't keep small mammals from eating them as whole, except by directing molecularly this chemical compound, capsaicin, so that the mouse can't eat it, but the birds are perfectly fine with it. And so I think this is another lovely story of chemical warfare, especially between plants and different types of small animals. Okay, so we talked all about the taste receptors and the gustatory ganglion. The gustatory nerve from the tongue projects to the brainstem through the nucleus of the solitary tract. It then goes up to the thalamus, just like it did for vision, and from thalamus goes to cortex, just like vision did. So this again is a canonical pathway where the primary afferents, or primary sensory neurons, project to the ganglion cells, and the ganglion cells, sometimes with a stop or two, go to the thalamus, and then from thalamus goes to a part of cortex. This is the same thing that we're going to see over and over again through all of the different sensory systems, with a couple of exceptions. And so food, unlike vision, for example, is something that a lot of us like. Like, I really like food. In my family, we always we like hot pot, and this is a tradition for us, especially on Chinese New Year. And so food, like lots of other sensory modalities, I think has this really special ability to elicit memories and nostalgia. And this is especially true of foods that are not necessarily appetitive to everyone. So this is a picture of a piece of stinky tofu and some blue cheese. Okay, I happen to personally like both of them. But again, I realize that some of this nostalgia is because I grew up eating these types of foods and I learned to like them, and they're not really all that. A lot of people don't like these types of foods, right? But for certain people, tastes and smells has a special ability to be deeply nostalgic, and you might smell a soup or something walking into someone else's kitchen and suddenly be transported back to your grandma's kitchen because it smells exactly like something that she would used to cook. And so I think this ability of foods and tastes and smells to elicit very powerful emotions and memories is something that is that's quite interesting in terms of how the neural pathways that underlie these powerful emotions and memories are. So there's a really interesting paper that I'm going to highlight here, just because it's something that I think is really interesting to bring this together between the sensory capabilities in the gustatory system and the ability for us to form these very powerful memories and the valence and the value that we assign to these different tastes and smells. So I told you that the gustatory nerve projects via some different waypoints, the thalamus, which then projects the gustatory cortex. The gustatory cortex has an organization where there's a part of it that's responsible for sweetness and a part of it that's responsible for bitterness. Sweet cortex and bitter cortex. Just like there was tonotopy in the visual cortex, where there's different patches of it that responded to different parts of the visual space, here in gustatory cortex, you similarly have this kind of topographical organization where sweet tastes are in one place and bitter tastes are in a different place. And you can see that they're anatomically distinct and you kind of separate them. Now, what this paper demonstrated is that if you look at where the gustatory cortex then projects to, the next place that it projects to a place that is called the amygdala. Now, I haven't talked to you about the amygdala before, so I'll just do like a 1 or 2 sentence summary here about the amygdala. The amygdala is a structure that is in the middle of your brain. It's a very deep structure. It's part of the limbic system, and we think of it as being associated with fear and memory and different types of aggression. So it's a very like primal feeling-oriented part of your brain. And what they demonstrated here is that the sweet cortex and bitter cortex actually project to different places in the amygdala as well. So the experimental methods they used here are pretty interesting. I'm going to take a moment to go through them with you. So what they did here is they injected a AAV virus, an adeno-associated virus, that is expressing CRE, and it's an enterograde AAV virus. Now, I'm going to break down those terminology. AAV, adeno-associated virus, is not actually an adenovirus. It's a totally different virus. It just has that name. So it's a virus that infects the nervous system. If you inject it into the brain, what's going to happen is it's going to get sucked up by the neurons. And this particular one is an enterograde AAV, which means that it's going to get sucked into the neurons and they get transported in the same direction as the axons. So in this case, if you're injecting it into gustatory cortex, it's going to get into all the cells in the gustatory cortex. And then all of those cells are going to then transport these AAV viruses in the same direction as the axons. Not backwards, forwards. That's what enterograde means. They're also retrograde viruses. This particular one is an enterograde one. So it's going to follow those axons and go to wherever those gustatory cortex neurons are going. And they're going to go towards the next place that they synapse onto. They're going to transport it all the way down the axon and hop to the next neuron that neuron was projecting to as synapses, okay? And because of the CRE system, they've engineered this mouse so that the CRE turns on at the next postsynaptic cell a GFP. Well, it's a red fluorescent protein. It's a TD tomato, so it's red, but it's like a GFP. It's a fluorescent protein that gets expressed whenever that cell is a postsynaptic partner of the cell through which you injected the AAV. So this is a kind of a viral track tracing here. It's kind of a really cool technology. So you engineer this mouse that has this property that the TD tomato, this red fluorescent protein, gets only turned on when it receives CRE. And you deliver the CRE not by actually delivering it, but by delivering it to a totally different brain area. And then so then only the projections that are between area A and area B receive that CRE and causes the cells in the amygdala, in this case, to glow red. And that's how they were able to demonstrate that if you injected AAV CRE in the bitter cortex, it goes to one part of the amygdala. And if you inject the same virus in the sweet portion of gustatory cortex, it projects to a totally different part of the amygdala, suggesting that there might be a possibility that these two different channels of information, bitter and sweet, can be associated with different feelings. Feelings is anthropomorphizing, but associated with the different ways in which we respond to stimulus that are sweet or bitter. And they further went on to demonstrate this with an optogenetic experiment. So optogenetics is it's a subject of the video. If you want to review the video on optical methods in modern neuroscience, there's a lecture where I talk to you about the basics of optogenetics, how it works. The short version is that we can turn cells on and off with light if we can get the right proteins in them. And so here what we're doing is we're going to, instead of having this Cree system, we're going to have an AAV that is carrying the channel rhodopsin protein. We're going to inject that into gustatory cortex, okay? And then instead of shining light at the gustatory cortex, we're actually going to shine light at the amygdala. And so it's only the cells that projected from wherever I injected my virus on that's projecting to the amygdala that are going to get activated. And what they found is that if they inject it into the sweet cortex and then they shine the laser in the amygdala, when the mouse is licking water, just water, the water is the same, it will lick more. Whereas if they injected the virus into the bitter part of gustatory cortex and shine the light on in the amygdala, the bitter projections in the amygdala cause the mouse to actually lick less, right? In other words, it's not the identity of the stimulus, whether or not the water actually was sweet or was bitter or whatever, but the fact that you're activating this amygdala limbic system area of the brain that made it so that the mouse actually behaved differently in response to the same stimulus. So it gets a little complicated, so kind of think about the logic here, right? So there's a way in which we identify different parts of the stimulus. The external environment, is it red, is it green, do I have emotional feelings about this, is it bitter, is it sweet, what do I do about it? And so in this paper, using this really clever technique of viral delivery of optogenetics targeted at different areas of your different neural circuits, they were able to disambiguate some of these ideas about how do you know what something is and what is the valence of that thing, how do you feel about it, what are you going to do about it? Okay, so that's something that's really clever. Okay, so key concepts to understand our sense of taste. We got really far, right? We went from a very basic taste receptor neurophysiology to some very modern techniques using optogenetics to dissect these rather complicated circuits, including our fears and our tastes. So we have taste receptor cells, and they're found in the taste buds. They are mixed and they do not localize the different parts of the human tongue. They don't have axons. They in fact activate gustatory ganglion cells just in the same way that in your retina, the photoreceptors activate retinal ganglion cells. So the gustatory ganglion neurons project to the rest of the gustatory pathway, including going through the thalamus and then going to cortex, and from cortex to other places unknown. They are the sweet and the umami and the bitter receptors are all GPCRs, which is hopefully not a surprise to anyone at this point. So GPCRs, this is not the last time you'll hear about GPCRs, because the next lecture is going to be about your sense of smell, and you will also not be surprised to hear that there are GPCRs in the smell system as well.